So our second speaker is also the main organizer of this event. Is Gergo Palfalvi. Gergo was working until recently in Japan at the National Institute for Basic Biology in Okazaki, and is currently now a postdoctoral associate at the Max Planck Institute. He's a fellow in the Institute for Plant Breeding in Germany, in Köln, in Germany. And today is going to talk to us about nuclear isolation from diverse plant species. Gergo.
Thank you very much, Marc. So my name is Gergo Palfalvi. As Marc said, at the moment I'm working in the Max Planck Institute for Plant Breeding Research in Germany. But most of the work I will show here was developed in Japan. Okay. As the title said, I will talk about nuclei isolation with from diverse plant species. And when I say diverse plant species, I mean very unconventional models or non-model species. For example, my main interest was leaf development and carnivory in the Australian pitcher plant Cephalotus follicularis. And we also worked with some other carnivorous plants to do single-cell sequencing. There are a lot of collaboration in my previous lab to use single-cell both RNA and multiome sequencing from Fismith Falcons, a moss model. And more recently, I'm working with more conventional model systems like Arabidopsis or tomato. So you can see there is a really wide variety of plant species with a lot of different considerations, and a lot of times nuclei isolation is the only way to set up these experiments, as protoplasting is not applicable or not set up for them, or we just have very time-sensitive experiments. And usually when you hear presentations about single-cell sequencing, this is the only slide you get about the data preparation. So people say that they collected some sample, they did either protoplasting or nuclei isolation, they did the 10x genomics pipeline, for example, here sequenced, and then you get the UMAP and all the results. Today, I do not want to talk anything about this part. I will only talk about the nuclei isolation, and I really want to bring you into the lab and talk about the different processes we did to develop the nuclei isolation protocols from all these plant species and how we ended up with the protocols we use today. So first, as Trevor already said, that you need to consider that you will use protoplasting or nuclei isolation, and there's a lot of differences between these two systems. The first thing people say is the RNA amount. Protoplast, of course, has much more RNA. It also includes the nuclei, so inherently it has much more. However, you most of the times get some cell type bias, as different cell types might protoplast differently or do not protoplast at all. And also, if you have a very time-sensitive experiment with, for example, a treatment, and you want to see like a 30 minutes, 1 hour change in your species, then a 2-hour protoplasting experiment will be very adverse for this. So for these considerations, from most of the plant species, we use nuclei isolation. And when you read a protocol, an article, for example, and you try to reproduce the nuclei isolation in your own species, in your own lab, this is the first thing you will see under a microscope, maybe, and it might discourage you, but I think you can be very positive because you can see some nuclei among the lot of debris and trichomes. So this was one of the first isolation I did. And I didn't give up after this, of course. There's a lot of steps we developed to remove all these debris. Of course, the trichomes can be removed very easily with some filtering, but there are some other debris. You can see starch, for example, a really big issue in some cases. And here I just show the basic process of our current protocol, which we use most often. So we have the plant sample, which we collect freshly on ice. And this is done as quickly as possible, of course. And then we chop the tissue with some razor blade, filter the sample, and then use fluorescence-associated nuclei sorting to get the nuclei cleaned up and enriched. And with different modification, I will talk shortly, you can get nuclei preparation without any centrifugation, which can be compatible with, for example, the 10x genomics single-cell RNA-seq input. So of course, also you can get the general recommendations from other places, like do not vortex your sample, use slow centrifugation speed, be extremely careful with pipetting, and if you can, use whiteboard tips, work always on ice and as quickly as possible. These are all true in these cases too. And this protocol will be shortly, in a few days, hopefully uploaded to the new protocols.io website for the plant cell atlas. And hopefully there will be much more protocols in the future. So if you would like, please screenshot this slide, or you can see it later, and you can visit the website to see shared detailed protocols for nuclei isolation and protoplasting. There is another method we started to use more recently, and this is based on the 10x Genomics nuclei isolation kit. We have really good results with it at the moment. However, I cannot confirm that with a totally finished experiment at the moment, but the nuclei preparation looks really good. Although 10x Genomics officially do not support plant preparation from these samples. As you can see, most of the steps are a little bit different. You freeze the sample in liquid nitrogen, then you disrupt with a plastic vessel. There's a filtering step after that also, and they use a column which is similar to an RNA or DNA isolation silica column. However, the nuclei should pass through this, and then with subsequent centrifugation, you can clean up your nuclei. So maybe in a few months, I can show much more finished data with this protocol too. So the first part is the sample collection. I mentioned fresh tissue on ice, and I also mentioned liquid nitrogen as flash frozen tissue. So which one to use? And it really depends on your species and your experiment. In our experiments, usually fresh tissues are much higher quality for the nuclei than frozen tissue. However, you have some limitation on the collection time, and we generally recommend to do within 30 minutes collection. Otherwise, your sample might be compromised for what you want to see. But with liquid nitrogen, you can alleviate this. You can store the tissue for short term. We would not recommend long-term storage. Usually, the longest one I tried was 1 week, and we already could see some degradation in the RNA quality of the samples in this case. But you can do, for example, a shorter term accumulation, like you freeze your sample in every 10-20 minutes, and you can collect much more samples. And when you have enough samples, you can just merge them and continue with the preparation.
Gergo, we have a little problem with your sound. It was difficult to hear what you said recently.
Can you hear now?
That's better. Yes.
Okay.
I'm facing the same issue again. Sorry.
Yes.
Maybe the video. I don't know. That's pretty awkward. Each time you start again, it starts to freeze a little bit.
Maybe I go away from the video. Is it better now? Okay. So just for the chopping, we just use a fresh razor blade always, and just chop on ice as quickly as possible until there is no visible materials left. And the idea is very similar with the plastic vessel. However, in that case, usually we use frozen tissue to disrupt the tissue. And a very important consideration is what buffer you use for this extraction. And we worked a lot on the buffer composition for the nuclei extraction to work with multiple species and have a minimal modification between species. So we generally use a low salt concentration buffer, which has low amounts of sodium chloride and magnesium chloride. Usually we exclude potassium chloride because we did not see any difference using it or not. We use 3-HCl for the buffering. And I think the most important thing is the detergents you use. People tend to use Triton X100, but this is mostly historical reason for plants. In animals, they tend to use Nonidat P40. And we found that Nonidat P40 or a very similar or identical detergent, Igapal CA630, works better for nuclei isolation. And this is the first optimization step we usually do when we have a new tissue or new species. We titrate the detergent concentration from 0.1 to 1%. The idea here is to remove most of the chloroplast or all the chloroplast with the detergent concentration, but do not have any effect on the nuclei quality. This you can see under the microscope later. We also include some small amount of twin-20, which does not affect the membranes, but it helps to remove the aggregations. And for the aggregation, we also use several, I mentioned here as anti-sticky chemicals like BSA and PVP, which helps to make the single nuclei suspension really single nuclei. And of course, there are some extra components like RNase inhibitors and spermidine. And the next cleanup step, there are several ways you could go. You could just do a quick and dirty centrifugation, which in some cases might work. I did work a lot with density gradient centrifugation and have a lot of data, so it works really nicely. But I would not recommend to do it because it's extremely difficult and very finicky to reproduce each and every time. But if you would like to know more about it, you can contact me and we can discuss gradient centrifugation. Our preferred method is cell sorting. I think we will also hear more from Dongbo later about cell sorting, which is a really good method. And the only thing I should say here is that it's really difficult to process, I think, more than 2 samples at once, as your samples should sit on ice while waiting the other samples to finish. And there is the column base, as I mentioned, from 10x speeds, but there is no official support, although there are some good results coming out at the moment. And for the fluorescence-associated nuclei sorting, we generally use a 70 micrometer nozzle, which allows us to collect nuclei at around 300 nuclei per microliter concentration, which can be readily used for downstream application. And we do this with DAPI for RNA sequencing. If we do multiome, for example, DAPI is usually not recommended, and we use 7AAD, and with multiple sequential filtering on different parameters in the cell sorting machine, you can get the really nice nuclei peaks. Another consideration you need to take care and talk with your technical staff is the sheet fluid. So the fluid which brings the droplets through should not contain any EDTA because it inhibits the downstream application. And with commercial sheet fluid, we did see a lot of reduction in RNA amount. So, and just see a few of the preloading quality checks. So I would recommend you to always check your nuclei before loading, even if you did the nuclei extraction 100 times. There's always some issue which can go wrong. And of course, you also need to know the concentration. So in this case, you can see some bigger chunks which are not optimal, but you can also see some smaller duplicates or triplets, which are even worse because it's much more difficult to detect in this resolution. And of course, you would like a single nuclei solution. Otherwise, your data would be compromised. So be aware of these aggregates, big ones and small ones, and try to avoid it. If you have these, maybe try increasing the BSA, PVP concentration, or work a little bit with the detergents. In the next slide, I show a bit lower concentration. There is no aggregates. There is a few low quality nuclei, which is not that nice, you can see. But also you can see, especially in the bright field, that there is a lot of non-nuclei small debris. And in this case, these are starch granules. And starch should be usually inert, so it should not interfere with most of the reaction. However, it can cause clogging in microfluidic systems. So you should take care to remove most of the starch also as you can. And gradient centrifugation, fluorescence-associated nuclei sorting are a really good ways to remove the starch from your sample. And then here I show a few good quality nuclei. When you zoom in, you can see the really nice nuclear envelope, nuclear membrane, very sharp. And you can also see a lot of cases, the nucleolus, which is a really good sign for a good nuclei prep. You can see also the DAPI. In some cases, it's not round, of course. So also you need to take care that your nuclei might not be round and it might be natural. So for example, this case, we confirmed that in the tissue, the nuclei also looks actually this eye or droplet shape, and it gives this shape after isolation. What you need to be careful is blebbing. When you see some smeared outgrowths of the nuclei, and in this case, you also can't see the nucleolus, these are lower quality nuclei. So if you have a lot of these in your sample, you might be able to sequence, but you probably will get a lower quality data. So if you have mostly these, then I would recommend to work with the detergent concentration to reduce it. And of course, you have the bad quality nuclei, which is mostly burst nuclei. You can see that there is no nice membrane anymore, and you can also see a more spiral shape in the duppy. So just remember that nuclei comes in all shapes, but the most important thing for them is the integrity. And with this, I would like to thank you, all my co-workers and collaborators all the years, and especially to Shoji Segami, Ruan, and Nan, who worked together to develop these nuclei isolation protocols. Thank you very much.
Thank you very much, Gergo. First two talks complement very one versus another with a focus on protoplast and the second one on nuclei. So that's really, really interesting. Thank you so much.